Monday morning. Yeah, it's a Monday. I get it.
Well, let me grab some tea.
Right, so Muge already shared the link, but let me reshare it so everyone has it. There you go. Okay, we lost Emily. Ah, there he is. Quick. Welcome to Neuroccino on this Monday morning, 9:30. Today we have Muge present a paper to us on interpersonal neuroentrainment during early social interaction. We're looking forward to it. Take it away.
Okay, thank you. Good morning, everyone. Let me share. Okay. Okay. How do you see my screen? Okay, so today I'm going to present this article, Interpersonal Neural Entrainment During Early Social Interaction, from this group in Cambridge, Sam Wass's paper. And this paper is basically about how interpersonal social influences, basically in this context, it's between a parent and an infant or between a parent and a child in the early life, how our brains respond to these kind of interactive interpersonal social influences. And so basically, they start by saying that when we are an infant, when we are a child, our waking hours are almost spent, entirely with our parents or with our caregivers. And this social factors influence how we learn, how we, develop our attention, how we learn things. And but most of the studies, when they investigate how these social interactions with our parents or with caregivers influence infants, they usually look at for example, infant's brain or the parent's brain, but not, they don't investigate the two brains interacting together, like what changes in the brain during that interaction. So that's why they say like it's important to use these hyper scanning techniques, so record from both brains at the same time. And it's also important to do these kind of scannings during like naturalistic interactions, so that the findings would be like naturally like ecologically valid. So we shouldn't restrain ourselves to controlled experiments, but do these type of experiments in naturalistic interactions. And then, so basically, when they look at different types of interaction measures, like between a parent and for example an infant, when they look at different interaction measures, they talk about ostensive cues. Because I'm not coming from this type of background, these terminology was very new to me. I don't know if you're familiar, but so basically ostensive cues are during an interaction, social interaction. Those are the signals that are sent to indicate communicative intent. And then they talk about contingent responsiveness, and these are social cues that show that you are sensitive, receptive to communicate, an interaction. So the both ways of the interaction, like from the view of the sender, from the view of the receiver. And then in the other part of the review, they talk about, so in the first part, they talk about these kind of social interaction cues. And then in the second part, they talk about neural entrainment. So if we start with the ostensive cues, so these are like, it's easier to understand with the the example right away. So direct eye gaze, for example, or child-directed speech, or like pointing to an object, for example. These are all different types of ostensive cues. And then when you, when you start an eye gaze, like direct eye gaze, or like point to an object, this means that you are, intending to start a, interaction, a communication, right? And so these type of ostensive cues lead to a range of changes in behavior. And then they lead to an increased behavioral entrainment. For example, directed eye gaze lead to an increased behavioral mimicry, or like mirroring facial expressions in infants. Or, for example, child, for example, what was the other example that I was going to give? Let me see. Yeah, so they talk about, like, similarly, like in language, vocal mirroring observed, when you start a child-directed speech, the child mirrors you. So these type of, like, mirrors your vocal, your vocal impression. So these kind of ostensive cues start some sort of like behavioral entrainment. And then and this type of behavioral entrainment later gives like some changes, influences on the neural response too. For example, during adult infant play, for example, directed gaze evokes larger cortical activity than, for example, averted gaze. Or, for example, child-directed speech, again, results in larger neural activation in infants compared to, like, adult-directed speech. So, child-directed is basically, it's like, that kind of speech with children is actually like seems to be useful. It evokes larger activation when you use child-directed speech. And okay, later on they talk about so in this first figure, it's a bit busy. It's a complicated figure, but what this figure basically show is that the different types of paradigms that are used in these kind of studies. So, for example, this is an example for joint play between a child and a parent, and then this sorry. And this shows a solo play. So they're sitting at a table together, but they are like separated by a screen, and then they are, individually playing. And then, for example, averted gaze, the parent looking away, directed gaze, parent looking directly at the child. And sometimes in these paradigms, they play games. For example, they play cooperative games or competitive games together. And, for example, basically they tag all the looks. For example, the child, when the child looked at the parent, or when the parent looked at the child, and then the times when the child was inattentive and the parent was inattentive, they have their EEG, like dual EEG, recorded at the same time, for example, in this case. And basically what this figure shows that during joint play, here on the left, look at the blue dots. So this shows the inattentive periods of the child is much less than the inattentive periods during solo play. So playing together with the parents helps the child to focus more, pay more attention. And, like, when you look at these red lines, for example, the up part is from the child, the bottom is from the parent. We see that during joint play, also the parent looks at the child a lot. So during joint playing, parents pay more attention to their child, for example. So this is, an example for, like, what kind of things they look in these paradigms, what kind of paradigms they use, basically. And then, okay, so we talked about ostensive cues, and then the other type of interaction cue is the contingent responsiveness. So this is not like from the point of the sender, like the sender of the signal, like the ostensive cues. This is contingent responsiveness. That's how the receiver, acts. So basically, this is like how the receiver indicates communicative sensitivity within an interaction by consistently responding to behaviors from the sender. So this shows that, okay, I'm receptive right now. I'm listening to you. I'm paying attention to you. So these type of cues are like mirroring, for example, when you listen to someone, you mirror their facial expressions maybe, or like marking with a smile or a nod, like that shows that you are receptive to interaction. And these contingent responses, so yeah, so these are also are known to be important for learning and attention in infants and children. For example, they say that like when adults presented word labels to infants contingently in response to their vocalization, for example, like when like by smiling, so the infant learns those word label associations better, for example, they say. And or for example, more contingently interacting partners in an interaction, they have stronger like action observer correspondence, they say. So what is an action observer correspondence? Basically it's for example, observing someone else perform an action involves neural activity in the observer becoming more like the neural activity in the person. So when you are not doing the action, but just observing someone, your neural activity will be similar to the neural activity who is doing the action. So this is kind of called the actor-observer correspondence. So people who are like partners that interact more contingently, like by more like showing that they're listening, have more of these, actor observer correspondence. Yeah. So, okay, so these boxes I skip. Okay, so yeah, so basically so these kind of like ostensive or contingent responsiveness results in some sort of behavioral entrainment between the interacting partners, right? They somehow like their behaviors, their mimicry, they mimic each other, their behaviors tend to become similar. But how do these behavioral entrainments affect the neural responses? So this part talks about neural entrainment. And then for neural entrainment, they look at two different things. So two different measures of neural entrainment. So one measure is concurrent entrainment. So when A is high, B is high, so also known as synchrony. So if activation in the brain of A, neural activity is high, then the activation in the brain activity of the B will also be high. So this is like synchrony. And then the other measure is the sequential entrainment. So changes in A are followed by changes in B. So they call this the sequential entrainment, but I think we can also think about this as like a causal, entrainment. Okay, so there are different ways of like measuring neural entrainment and like doing these like hyperscanning or hyperscanning kind of using hyperscanning methods. The most widely used ones are the EEG and FNIRS. So if you're measuring neural entrainment from seconds to minutes scale, so with a lower temporal resolution, then the functional near infrared spectroscopy is used. And then this is, I think, widely used because it's like portable system, not heavy, like it's easy to do this type of experiments set up with the infants. And so basically with using FNIRS, they would like, they can measure concurrent and sequential entrainment by calculating the cross correlation in this deoxyhemoglobin levels between the, two partners' brains. And the other technique is widely used technique is EEG. So this is for measuring sub-second scale entrainment. And usually the most commonly studied, bands, activation bands, frequency bands are theta and alpha, because these frequency bands are associated with attention and learning. And when analyzing EEG, there are a lot of like signal processing techniques to measure entrainment. So if you're like measuring concurrent entrainment, so when A changes, B changes at the same time, to measure this type of synchrony, then you can use like phase locking value, so which is basically like measures phase coherence. So how the signals in one brain oscillates together with the signal from the other brain. So how they fluctuate, oscillate together. And the other technique, if you're measuring not concurrent but sequential entrainment, so the changes in A affects B, changes in B. So then you would use like techniques like Granger causality based techniques in EEG analysis. So they say, okay, there is neural entrainment. You're measuring like neural entrainment, but how is this possible? How two independent brains can be neurally entrained? Like, what is the mechanism for this? And then for this one, they talk about basically phase resetting. So phase resetting is, for example, when our brains when an external event affects the activity in our brain. So so I will explain this with a figure. So yeah, here in this figure, for example, they show a signal measured from a child brain and signal measured from an adult brain. They're doing their own thing, and then there's a phase reset, and then the both activity in both brains are aligned nicely. So now they're in synchrony. So what is this thing that align the activities? It's usually like some sort of ostensive cue, like a gaze or like a speech onset or a touch, for example. That would, like when two partners are like dyads in a dyad are interacting, these type of like gaze, speech, touch, these can reset the activity, neural oscillations in both brains at the same time, putting them in entrainment, in synchrony, basically. And then the idea is during when the two brains are like entrained, this creates a high receptivity periods like between the partners, and then when the parent at that time say something like this is a ZERP, then the child is more attentive or learns better. So yeah, so basically yeah, so this is like the mechanism of how suggested mechanism of how neural entrainment between two brains are achieved, basically by phase resetting. And wait a second, they also talked about something else here. Sorry. And in addition to phase resetting, they say that actor-observer correspondences, just like watching someone do an action, will create similar type of activity in the brain of the observer. So this can also be a mechanism of neural entrainment. So just watching, observing someone do an action. And in the last part, they talk about, okay, what is this useful for? Like, why? I mean, entrainment between two brains, why is it useful? And does it have any role, actual role, in learning and attention? Okay, so the first the one role that one use I already mentioned about, so in this figure. So if the two brains are entrained, so this creates a window of like high receptivity, then this affects how we attend, how we learn. And then the second use of this neural entrainment, they talk about allostatus. To be honest, I'm not super clear about this one, but in my understanding, so allostatus is it's a key in social interactions, basically helping the partners maintain a stable state. So this is mostly for the parent helping the child to maintain a stable state, because children, like infant's brain is like very stochastic, and then it's just responds randomly. And by neurally entraining to a grown up, to an adult, their brain is somehow like trained to function more like maturely, in my understanding. They talk about in one of the boxes, they talk about the different ways how this kind of allostasis is achieved between a parent and a child. For example, if the child has a high arousal level, usually the parent approaches the child with a lower arousal level, so that the child gets lowers their arousal level too. And or if the child, for example, if a parent is depressed, for example, they cannot respond to their children's like arousal levels properly, and then cannot help the child to maintain their stabilize their states. So find the allostatus. But as I said, I'm not very clear about this part. It's very the terminology, and it's very different than what I'm used to. So but yeah, they talk about this too. And yeah, I think this is all I prepared. And they also talk about some outstanding questions in this field of research. But yeah, I think, yeah, for the sake of time, I stop here.
Lovely. Thank you, Mege.
Mhm.
Any questions? Not yet. Ah, Eduardo, here we go.
Well, I don't have much choice. Thanks, Muge, for presenting the paper. Yeah, so maybe one of my questions is relating maybe to one of the last paragraph that you already mentioned, but I was still wondering, like, what kind of argument they provide for to kind of like to argue that these like all measures that you can extract from brains are actually more like on the side of being a cause of the behavior rather than just a consequence of the so say for example, phase resetting, how much is it like a consequence of things happening in the brain rather than a cause for whatever attentional and learning phenomenon you can observe.
Yeah, they talk about, for example, when the two the parents and the child are in the same environment, is it really that their brains are getting entrained to each other, or is it just like something in the environment at the same time affecting their brains at the same time, so that it looks like the two brains are entrained? So that's that's a valid question. So in these
Because
experiments,
just because things are happening at the same time in two brains, that doesn't really guarantee any kind of
yeah.
causality.
Yeah, that's true. So, uh, Yeah, I don't know how they deal with this issue. So how did they probably have some other sort of like control conditions, but this is actually mostly a problem during like naturalistic paradigms that they encourage here. I think they use some sort of like bootstrapping techniques by like shuffling the trials recorded, shuffling all the trials or phases recorded from one brain, like and then repeatedly, and then um, comparing the actual brain activity to this distribution um, to see if it's real or if it's just by chance. I don't know. Yeah.
Yeah,
But
no, it was just to be curious if they spend maybe a bit more on that. It's something we can dive a bit more on. And also, if I have can I have another question?
yeah.
So this is more maybe related to how useful is hyperscanning, because like especially if you're doing it with adults and kids, then you're kind of relying on the assumption that the brain activity is similar in like statistical terms, like but we know that like the activity of a child's brain or the activity of an adult brain is, well, there is a lot of stability, but it's also like there is some differences, right? If you take the EEG signal, or like the 1/f structure is different. For example, I mean, just as example. So how like how valid is
Yeah.
the assumption of comparing like same measures for brain activities that might have differences?
The only like as far as I remember, the only thing that they mentioned is the child brain and the adult brain have different dominant frequencies. So being like it's alpha band in an adult's brain, and it's usually the theta band in child's brain, in infant's brain. So they the dominant oscillation frequencies are different. So to look for pattern similarities between these different frequency bands, they would use like cross frequency coupling. So techniques to compare them. So they would compare alpha in adults with beta, theta in infants.
Okay.
That's the only thing that they mentioned as far as I remember here.
Okay, interesting. Yeah. Well, one could ask other questions on that, but yeah, I'll leave it to.
Well, fine with me. If
Yeah, no, and it's just
yeah.
yeah. Thank you, Miguel.
Yeah. Okay.
Marcela.
Yeah, thank you, Mush.
Yeah.
I'm just wondering, I'm not sure if they consider, I know it's a review, but how the social interaction change across age. I don't know if they are talking about a specific group of kids, specific age, because the social interaction is different when they are more baby compared to when they are older. So I don't know if it's something that they consider. Also, some other social aspects or the context, the environment.
Yeah. Yeah, so I think only in the outstanding questions they talk about like how one of the outstanding questions is how does interpersonal entrainment changes over development? So they only talk about this in the outstanding questions, but they don't target this question in this paper. Most of the examples, like citations that they give, is for infant studies to until age 1. So yeah, they don't talk about more like toddlers or children, But yeah, I agree. Valid question.
Thank you.
If I can bring us slightly back to what Eduardo asked earlier about the causality and the mechanism. So I was also wondering if the synchrony that you see may be epiphenomenal, whereby they're responding to the same cues in the environment rather than to the interaction between them. In a sense that if you see the same thing or if you hear the same thing, you could have similar brain signals without requiring any interplay and communication between the brains. But that then also led me to the question about the mechanism. So obviously with the techniques they're using, there is no spatial resolution in the data that's really reliable. But and that's a general hyperscanning question. What's the underlying mechanistic understanding of how our brains synchronize, if they do? Like, how does it actually work if it was really brain-to-brain communication rather than an external cue that triggers the signals? How?
Yeah, so for the mechanism, that's the two things that they mentioned is this phase resetting, like something that happens, resets both brains, and then put them in synchrony, put them in entrainment, that phase resetting. And the other one is basically it just says what, watching someone, listening someone will like if the infant is watching the parent or listening to the parent, or like, it would create similar activation in the infant's brain to the parent's brain. It's like mimicking, like mirroring, basically. Neural mirroring, I would say. But yeah, that's like being in the same environment affects, the visual or auditory stimuli and the environment affect the two brains at the same time. That's really valid. I don't know, but I really don't know if they talk about how to eliminate this possibility. Yeah, shared external perturbation.
I mean, it's a general
Yeah, they
scanning.
just generally mention that, but they don't talk too much about how to, eliminate this possibility of this external perturbation affecting both brains at the same time. Yeah.
Don't they have maybe, I have no idea about like how hyperscanning is done and what are the experimental paradigms, but I would expect to have like some sort of control condition when you do things together and you record brain signal at the same time, and I don't know, some sort of like isolated condition when you do hyperscanning, but maybe I don't know, the two people are like in different rooms or like doing a similar task, but without the behavioral interaction. And then you look for differences in the brain, because then if you find the same synchrony or whatever measure you're looking at, and
Yeah.
in both conditions, then it's not really an interaction.
But I just don't know how those control conditions would look like. But one example they gave for let me see if I can find like yeah, so adult participants, for example, listen to a real life auditory narrative compared to a temporally scrambled version. For example, in this case, listening to the same narrative, they listen to the same auditory stimulus. So both partners get the same auditory stimulus, and then you may think that their brain would be entrained to that auditory stimulus separately, but at the same time. But then they find that when they use the like temporarily scrambled version of the same narrative, so it's also in this condition, again, they are listening to the same auditory stimulus, but the neural entrainment, the neural synchrony between the two brains in this, when they listen to the scrambled narrative was lower compared to when they listen to the actual narrative. So then they say here that this they have a shared understanding of the story when they listen to the story compared to when they just listen to some jibber jabber,
What do
So
you,
that's
yeah, sorry, just one thing, like the scramble version would be the same for each one, or?
Yeah, they listen to the same scrambled story. Yeah.
But then it just means they are both confused by the same scrambled version, no? Yeah, it's a bit strange, like the interpretation, but it always is. But yeah, okay, interesting. Yeah, but yeah, thank you.
Under, quick question.
Yes.
Thanks very much for the presentation. Really
Yeah.
interesting.
Mhm.
I'm wondering on child-directed speech. You mentioned that it could be beneficial,
Mhm.
and even though this is a bit of a topic of debate, especially towards the speech part, I wonder if it's more than that, and it's not only like what is being said and the message, but maybe the benefit could stem about the more obvious gestures and the ostensive cues that were mentioned. And I don't know what you think about this.
I don't know the exact experiments. Probably they would, control for these kind of like gestures, but just maybe look for acoustic differences between child-directed speech and adult-directed speech. Because, for instance, the basically are different. Child versus adult-directed speech is different in terms of pitch. So child-directed speech is usually higher pitch, but also I think I actually don't know very well about child-directed speech, like what is considered as child-directed speech. So, but in my understanding, like pitch is the main thing, and also like slow. For example, when you speak to a child, more like you articulate more clearly, speak slowly. Maybe also that's why the speech rate is also different, maybe. But yeah, if they have this, visual part to maybe like you smile more, like articulate more openly during child-directed speech. But yeah, so yeah, I don't actually know about the experiments from these different studies that cited, if they are like only auditorily listening child-directed speech or just seeing someone. So what are the balloons?
That's the cue to wrap it up.
Oh, okay. So yeah.
I don't know, you must have had a gesture. Thanks so much. There's clearly plenty more we can discuss. Happy to keep the conversation going after, but it's time to release everyone into their week. So thanks for presenting the paper. Just a quick note, we have a CNS talk this week on Wednesday, 4 p.m. by Nele de Meyer from Oxford on can routine clinical imaging data help unravel post-stroke cognition. So I hope to see you there.